everyone, I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, today we are going to know about lip prints. Lip prints, we all know these are the marks which can be left from the lip at the crime scene. We will study how these lip prints can be left at the crime scene, how these lip prints can be collected and can be analyzed in the forensic science laboratory. Firstly, we should know the history about lip prints, how they were identified as an evidence for the forensic investigation, their collection from the crime scene as well as from the suspects, and the analysis of lip prints. The science of lip prints is known as chiloscopy. Chiloscopy is the study of lip prints, which is an ignored but a very useful area of forensic anthropology. Forensic anthropology, if we talk about in a broader term, is an area where we use this science to identify the human body. Maybe it is a dead body or a living body. So lip print is one of the evidence which were first used for identification in 1950s that stimulated the research scientists in the next 2 decades, leading to the acceptance of this technique as an evidence in criminal justice system. The external surface of the lip, the prints has many elevations and depressions, forming a characteristic pattern called the lip prints. The external surface of the lip print is characterized by patterns of fissures and grooves that are genetically determined. So like on our fingerprints, we have grooves and certain patterns which are being transferred to other surfaces when our fingers come in contact with other surfaces. Similarly, these patterns which are present on our lips on the external surface of the lip area, they get transferred to the places when it comes in contact with other places. Now, at the crime scene, there are many cases where the lip prints can be found. They can be found mostly in the cases of sexual assaults. They can be found when there is a fight and the person his lips get touched to any surface, whether it's glass, wood, etc. They can be found if any criminal or suspect they ate something or they drink something out of any bottle or glass. So the lip prints can be found on those surfaces. Now, how this evidence can be used for the examination? Any evidence which is important in forensic science should be unique and should have certain characteristics. So, lip prints being uniform throughout the life and characteristics of a person can be used to verify the presence or absence of a person from the crime scene, provided there has been any consumption of beverage, usage of any cloth, tissue, napkin, etc., or left of lip print marks at the scene of crime. The studies have also observed that the twin possesses a very similar lip patterns. The forensic significance of the lip prints are they can be present at the scene of crime, especially in rape cases. In burglary cases, lip prints can be found on drinking utensils, over glass, cold drink, cans, bottles, etc. Lip prints can also be present in hit and run cases on the vehicle. They can be found in cases where fighting occurs between two persons, and it could be present on the glass of the window, on door, table, glass on a tape, envelopes, etc. Now, if we talk about how these lip prints were identified as an evidence and who studied it first. The pattern furrows on the red part of the human lip was first noted and described by the anthropologist R.S. Fisher in the year 1902. They are similar to fingerprints, but they are very different also. Lip prints and fingerprints both are means of identifying criminals. Like our fingerprints, we have certain patterns on the tips of our fingers. We have grooves, fissures. Similarly, on the lip prints, we have certain patterns and which can be helpful in identifying an individual because they are individualistic in nature. Different persons have different lip prints marks. The use of the lip prints was first recommended as early by E. Locard, that is Edmond Locard, in the year 1932, who is one of the France's greatest criminologists. Locard, he also gave us the principle of exchange, that if something comes in contact with any other thing, it leaves an impression. So Locard has studied many impressions during his tenure. He has seen which evidences can be used to identify an individual and which are unique to any person. Dr. Martin Santos, he presented his work on lip prints in the 4th International Symposium of Forensic Science Medicine, which was held at Copenhagen in the year 1966. So the scientists, they have studied lip print patterns, and then this evidence was identified as one of the evidence which can help in identification of an individual, as it is unique in nature. On the basis of various researches done in the field of lip print, this is suggested that lip prints evidence is suitable for the successful comparison, analysis and identification of a person to a crime. Professor Suzuki had studied lip prints in 18 pair of identical twins, and they have found remarkable similarities of lip prints among the twins. We have to notice one point, that there were similarities in the lip prints among the twins, but still there were some differences found. So if we can say that DNA of two twins matches, monozygotic twins DNA is same. So we cannot identify two twins on the basis of their DNA. But on the basis of lip prints, they can be identified. However, the studies have shown that identical twins in which lip prints may differ also. So there is an interesting aspect about the uniovular twins, which are the monozygotic twins, that they are in fact the same individual biologically, their cells have the same protein, the same genetic information and so on. Thus, if one of the uniovular twin commits a crime, the other could be implicated even if DNA profiling is used, because they have similar DNA. So, for the uniovular twins, if they contain the same DNA, they can be convicted for the same crime. If one person has committed one crime, the other uniovular twin or the monozygotic twin can be held for the same crime, but they have very different lip patterns. Similarities can be found in their lip patterns, but moreover, categorically they can be divided in different categories and they can be individualized on the basis of their lip prints. Now the anatomy of lip prints, if we say the middle part on the upper lip is known as philtrum, then we have the upper lip and the lower lip, and where both the upper lip and lower lip combine, that part is known as vermilion. Upper lip, we can also call it as, in scientific language, labia superfluous infetata. It is the strip of the smooth skin that borders the upper edge of the mouth. It is usually thinner than the lower lip and can have pronounced peaks to either side of a groove at the center of the mouth. Lower lip is known as labium inferius, is the strip of smooth skin that borders the lower edge of the mouth, and it is usually wider and vertically deeper than the upper lip. So, if we look at the anatomy of the face, the mouth on the upper side, the portion is called the upper lip and the lower side is called the lower lip, and where both the upper lip and the lower lip combines, that portion is known as the vermilion. Vermilion is the border of the skin of the lips and the surrounding skin of the face. In some individuals, this is more pronounced, while in others, the skin types seem to almost blend. Then the next is the cone-like structure on the upper lip, that is known as the philtrum. It is the groove-like indentation often found at the center of the upper lip. It is responsible for the biggest difference in shape between the upper and lower lips and thus becomes more pronounced when the mouth is puckered. Now, whenever we use any evidence for analysis in forensic investigation, we should look why that evidence is unique and why that evidence is individual. So, the lip prints, when they were identified as an evidence in forensic investigation, because they do not change throughout the life, they are as unique to the individual as palatal rugae patterns. Similarly, like as fingerprints, they are useful for personal identification. Lip prints can be both type like fingerprints. They can be latent and they can be visible. If there is any lip mark, lipstick on the lips, then they can be visible lip prints. If there is nothing on the lips, then it can be latent prints. Like fingerprints, lip prints can also be developed and lifted. So, lip prints can be classified into majorly 5 types, through which they can be categorized into different categories, thus which help for further analysis. Type 1 is full vertical grooves. Like in image we can see, there are vertical lines on the upper lip as well as on the lower lip. This full vertical grooves compiles the full vertical classification. We can call them as vertical fissures as well as vertical patterns. They are from end to end. If we can see, they are bigger in nature. We can see they start from the upper lip top area to the middle, similarly in the lower lip. Like full vertical grooves, we have short vertical grooves. They are similar to full vertical grooves, but they are incomplete vertical fissures, where you can find only small thin lines, just like the vertical grooves. Type 2 is branch grooves. Branching grooves is one of 5 basic types of lip prints. This type of lip print patterns is where both vertical and horizontal grooves are present. This classification of lip print is given by Suzuki and Tsuchihashi in the year 1970. So, they appears as they are combination of vertical and horizontal lines, they appear as branching grooves. Type 3 is known as diamond grooves. This classification was also given by Suzuki and Tsuchihashi. It is one of the 5 basic types of lip prints, wherein the characteristic patterns present diamond shaped grooves in the lip. The pattern is kind of crisscross in nature and it appears to be like a diamond. So, they have classified this print as diamond grooves. Then we have rectangular grooves. Rectangular grooves is the type 4 also known as, which is one of the 5 basic types of lip prints, wherein the characteristic patterns present grooves on the lip resembling a net-like design. So, the branching also we have vertical and horizontal lines, but they appear more in the form of branches of a tree. But in the rectangular grooves, they appear as the horizontal lines which cut the vertical lines. So, that is why this type of classification is known as rectangular grooves. Type 5, all other patterns, those are irregular, non-identifiable patterns. They all come in the type 5. They are all the mixed pattern. They may have the branching grooves also. They may have rectangular grooves. They may have diamond grooves. So, the combination of all the different types, they come in the type 5. Now, on the basis, whenever we have some print from the crime scene, we can see in which type it falls into. and then we look for the individual characteristics which are present in the lip prints. So the next is, after the classification, how the lip prints are collected from the crime scene and how the evidences are collected from the suspects for the comparison. So there are two ways of collection of lip prints: the collection of visible lip prints by applying lipstick over lips, and the collection of latent lip prints by using black magnetic powder and fluorescent powders. So for the collection of latent prints, this is the collection where we are taking lip prints from a suspect, because at the crime scene, whenever we have a visible print, either we will photograph it, or we can taping method we can use, like for fingerprints identification, to tape up the fingerprint and to paste it on a white paper. But for taking the prints from a suspect, we have to take a plastic slide and a container. The exterior of each container is marked with the individual's name, and we ensure that the slide we place our lip prints goes back into the container marked with the name of the person whose lip prints are present on that. The plastic slide on one end with white tape is kept in the container. This other end where the white tape is present, it should be used to pick up and remove from the container. We should not touch the slide or the plastic thing at any point, because it might leave any other print on that plastic slide. We can use a clean soft towel to wipe it, and we should not use any abrasive fabric, as this will scratch the surface. This slide is then placed on a clean, flat, smooth and solid surface, so that the side with the plus sign located on the end with the white tape is always facing up. Now make a lip print on the slide, and if the lips are dry, then chapstick can be applied on the upper lip, that you will use then to rub with the lower lip, and one application will probably be too much and the lines on the print will be filled in. It is suggested that the first print should be made on the piece of a glass and then the slide, so that the lip should not be, there should not be any merging of the lines. When you have the print on the slide, place the slide back into the container, ensuring the print portion is not pressed up against the wall. Otherwise, the print which is being given by the suspect will be wiped away or smudged. So that was the case for the fingerprint taken, the latent fingerprint taken from the suspect. And now how can we develop this latent fingerprint, which can also be found at the crime scene, maybe on any glass material or any beverage which the suspect might have consumed, or any glass surface, or in the assault cases on the skin of any person or any clothes. So in such cases, then the magnetic powder should be used for the identification and for the development of this print. This black magnetic powder is similar, which is used in the case of fingerprints. For the development of prints, the camel brush is used with the magnetic powder to rub it. The prints are being developed, and we can see that with the use of black magnetic powder, the prints are being developed on the plastic sheet. Same way, when we found the latent fingerprints on the crime scene, as we look for the fingerprints which are invisible to the naked eye, so they are called as the latent prints. Similarly, we search for the lip prints, and wherever we found these lip prints, they can be developed with the help of magnetic powders. And after the development of these prints with the help of magnetic powder, we can lift them up, and those can be stored in any container. So this was the method which we were discussing, that was for the latent prints. For the visible print, we directly use the lipstick. This method proved to work better because we have the clear prints. So easiest method is to use a lipstick. one coat on both the lips, and the best method is parting the lips slightly so there is a recognized separation between the two lips. We usually make a raised thick book on a desk which works well so that the nose can be on the one side and the lips can be directly on the book. So the lips need to start from one corner of the mouth and roll to the other corner of the mouth, all the while maintaining contact with the paper. And after taking the prints, write the name of the person on the upper right of the paper, or two prints can be taken, one on the upper half and second on the lower side, and in the middle write the name of the person. Now, after collection of prints from the crime scene, for the whether it is a latent print or it is a visible print, the forensic scientist has to analyze this print in the forensic science laboratory. We always divide the print into four parts. From the center of our upper lip to the center of the lower lip, we can put an imaginary line vertically, and from the left of the lip to the right of the lip in the center, we can put a horizontal line. So we have the upper left and the lower left, and the upper right and the lower right. For these four categories, we look for which all different patterns are present in these four categories, like whether the classification is type 1, which was our the type 2 classification and type 1 classification we have, or either we have the diamond classification, branch classification, rectangular classification, or the mixed classification. So depending upon what type of patterns are found in each quadrant, we can call them the first quadrant, second quadrant, third quadrant, and the fourth quadrant. We look for which patterns are present in the four quadrants. We look for all different patterns. There could be mixed patterns in one quadrant. So on those basis, we can see and match the different types. Let's see in the upper left, if they are saying that type 2 was present, which was a branched pattern with the type 1, that is the vertical fissures. In the upper right, there was type 1, which could be the vertical or small long patterns, or type 2, which is a branched pattern. Type 3 was a diamond pattern, and type 1 again is a vertical pattern, and in the lower right we have type 2, which is a branch pattern. So similarly, depending upon which kind of patterns falls into this four quadrants, we divide our prints according to that. So all the prints which are found at the crime scene, which can be collected, the latent prints which can be collected using the magnetic brush method. Firstly, we have to look for the places where these prints are present using UV light, or mostly they can be seen on the beverages, on the glasses, or any bottles, anything which is being touched by the lips of the criminal or the suspects. Or in the sexual assault cases, we have to look for the clothes of the persons where these lip prints can be found. So these prints which are found at the crime scene, the latent prints which are developed by the investigating officers at the crime scene, then they are taken to the forensic science lab, and as we don't have any patterns or any database of lip prints, so we have to look for the suspects and we have to take the prints of the suspects. We have previously discussed two technologies, one of taking latent prints and another one of taking visible prints in the cases of lip prints. Depending upon what kind of prints are found at the crime scene, it is better to take the same kind of prints from the suspects. Supposedly, if you have found latent prints at the crime scene, then you should take latent prints from the suspect, and if you have found visible prints from the crime scene, then you should take control visible lip prints from the suspects in the laboratory. As we analyze for the fingerprints, similarly we analyze for the lip prints. In the cases, we divide into 4 quadrants and then we can analyze it. So to conclude, I will say, while the lip prints have the potential to be a unique and individualized form of forensic evidence, their use is not as widespread or established as other forensic techniques. So the field of stereoscopy is still evolving, and further research and validation are needed to establish the reliability and admissibility of lip prints in forensic investigations. Thank you.